Our next speaker is Jane Lipkowski from Regenerative Patch Technologies.
Great, thank you very much. It's a pleasure to be able to introduce you to Regenerative Patch Technologies. This company is developing a composite cell therapeutic and membrane patch to be used for the treatment of macular degeneration. So RPT, Regenerative Patch Technology, was formed in 2011, and it is essentially, it's located in California, but it's been formed to commercialize a product that is being developed right now for the California project to cure blindness. And that is comprised of many different organizations, but in particular, USC, where the project is led by Mark Humayun and David Hinton. Mark Humayun, who has been most recently a founder of Second Sight, and recently had a approval, CE mark, and a pending BLA approval for a neuroprosthesis device with Second Sight. Also important to the project are the folks from UCSB, Dennis Clay and Link Johnson, who are very involved with developing the processes for manufacturing the RPE cells. And this membrane technology, which I will be talking to you about, is really been developed at Caltech, and the manufacturing, the final GMP manufacturing, at least for the phase 1, will take place at the City of Hope. It's really important to mention and thank CIRM for funding the Disease Team Grant, which was awarded over 2 years ago now to develop this product. So what is the product? The product, again, is a composite product. First, it contains a Parylene membrane. This is an ultra-thin, diffusible membrane that essentially represents the Brooks membrane. And it's important to have this Brooks membrane, especially if you're thinking of looking at patients with geographic atrophy, because it's often that this Brooks membrane is significantly damaged. And it is important to look at recreating the boundaries of the tissue architecture in the retina and the subretinal space to actually recapitulate what is normally found in the retina. So this is a diffusible Parylene membrane that, again, mimics the diffusion capabilities of the Brooks membrane. Plated on top of this membrane are RPE cells. They're derived from human embryonic stem cells, and they're polarized. They're to provide the both apical and basal functions of RPE cells that are required to distribute and to eliminate waste from the photoreceptor layers, and to also interdigitate and provide vascular support for the photoreceptor layers. This is just a picture here, electron micrograph, showing the Parylene substrate along with the mature RPE cells that are plated on this membrane. And again, this is designed to recreate the architecture of the basal retina. So in this particular case, the Parylene membrane providing a support and separation from the chorion capillaris, and providing support for the RPE cells, which subsequently provide support for photoreceptors. So this architecture of the tissue is really important for the disease that we're looking at treating, in particular the atrophic form of macular degeneration, and more particularly more advanced stages looking at geographic atrophy. So what you see in this condition is that there's a loss of a retinal pigmented epithelium, degeneration of the Brooks membrane. If it goes far enough, you'll actually see photoreceptor layers. There's often an accumulation of drusen. So we're looking at treating patients with the dry form of macular degeneration, and in particular looking at geographic atrophy. So the design of the product is really to look at addressing the pathology that is in the retina. As you've heard from previous speakers, that this market opportunity is enormous. There are about, just with geographic atrophy, there's a little bit, about 1 million patients already. The prevalence is about 1 million patients in the US alone, and that is expected to go up by 50% by 2020. And we're looking at, in this conservatively, over $1 billion market. So we're designing the product attributes of this particular product, really taking into consideration a lot of the different topics that we heard discussed earlier this morning. Obviously, this product needs to provide functional support for photoreceptors, provide improvement in visual acuity, and be implanted using simple procedures that could be used in the outpatient clinic. And under these circumstances, I will show those to you a little bit later. It's an off-the-shelf product. It obviously needs to be introduced safe and efficaciously. And the plans are at this particular point to use none or local immunosuppression. It is a scalable process that can supply the demands for the very large market size. It's a stable product that can be stable for at least a year. No complex processing at healthcare providers, because that is extremely important in terms of getting a market adoption of this particular product, and obviously having COGS compatible with reimbursement policies. So we're looking here at just pictures of the various components of the patch, of the implant. First, there's the Parylene membrane. It's made out of a Class 6 biocompatible polymer that's been used for 30 years in implantable devices, medical devices. For instance, used to coat catheters, used to coat electrodes. It has a permeability very similar to the Brooks membrane. It has a thick Parylene mesh on the outside and in between these circles to provide some rigidity and mechanical support so that you could introduce these safely. But also it has an ultra-thin membrane, which is these various circles that you see here, which really provide the diffusional capabilities and characteristics that are similar to the Brooks membrane. The RPE cells are 99% pure. They carry out the expressive genes and carry out many of the functions, for instance, phagocytosis. I'll show you some pictures later. Phagocytosis, which is very critical for the function of RPE cells. And then there is, in fact, the composite, where these RPE cells are plated on the membrane, and you can see the final product. The membranes are manufactured at Mini Pumps, a group that is well established in making under GMP medical devices such as these in membranes that can be used, made from Parylene. The cells are manufactured at the City of Hope for the phase 1 clinical studies, and the final product will be assembled at the City of Hope. Why are we interested in polarized RPE cells? Why is that important? Well, you can see here that there are all these villi that form as a result of the maturing process. These villi are very important in interdigitating with photoreceptors and allowing for that cross-contact for all the various functions of the RPE cells. You can see that they produce, when they're polarized, they produce more PEDF, which is important for photoreceptor support. And they also, at their basal surface, provide VEGF, which is in fact controlling the vascularity of the vascular system that is below the Brooks membrane. We know that these membranes now will survive when we implant the membranes with the coated RPEs into the subretinal space. We can see that here is the membrane right here, this opaque area, this white area. And on top of it in brown, you can see the RPE layer that is implanted in the subretinal space. They are human cells. In this particular case, you can see they stain red. They are also RPE cells and their composite function. We see that their survival, when you look at late-stage animal models of macular dystrophy, we find that when we implant them using these membranes as opposed to suspension cultures, that we see much superior survival. And in fact, in this case, we're looking at 2 months post-implant, but that we see essentially no survival in these late-stage models of macular degeneration. We also can see that these cells, when implanted on the membranes, phagocytize rod outer segments. So for instance, if we're looking at, in this particular case, the rhodopsin positive in red outer segments, and you can see here's the Parylene substrate with the RPE cells that are located on top of that substrate. And if you look very closely, you can see these red dots, which are the RPE cells, in fact, phagocytosing the rod outer segments. So they're functional. And finally, looking at an animal model of macular degeneration, what you find, in fact, is that they, in terms of their behavioral activity, head tracking to the right and to the left, you can see that animals that have received this particular implant with the RPE cells on the membrane, in fact, has superior head tracking ability. They eventually decline, but as in this particular case, the animal model, they develop vascular problems, and the model has limited utility after about 24 to 26 weeks. So you can see in this particular case, use of the implant with the RPE on the membrane does lead to better head tracking ability. So you might be asking, how are you going to deliver this membrane? And the membrane is going to be delivered through an outpatient procedure. In this particular case here, we're seeing, we've done all the modeling for the implantation of this in the pig, in this case the Yucatan pig. Here we're looking at the membrane. You can see the membrane with the pigmented RPE cells right on top of the cornea. We've developed a tool right now that can grab this particular membrane, and you can see it somewhat here going up into the injection device. It curls it up and puts it into a little roll that can be subsequently implanted into the subretinal space. Here you're seeing the tool making a small retinotomy to insert it into the subretinal space. After implantation, you can see by OCT here that there's the membrane in place. So it is an outpatient procedure. It is a procedure that will, there is precedent for this particular procedure, and what we've done is really modified and made this particular tool to facilitate making a very small incision to get this implant into the subretinal space. We've been working now with the team to establish timelines. We've had our pre-IND meeting and are executing the work in order to start our IND enabling studies and to enable clinical trials that we believe will start in late 2014. We're looking at targeting geographic atrophy and also some orphan disorders of the RPE that are unique from some of the other orphan disorders that various other organizations are examining right now. So in terms of intellectual property, RPT has an option to an exclusive license from from USC, UCSB, and Caltech to all the various technologies that are going to be required to commercialize this particular product. We have licenses now to the ultra-thin parallel membranes, the RPE cell growth and differentiation conditions, the growth on the polymeric substance, the instrumentation, in fact, to insert it into the eye, and also looking at methods of cryopreservation of this particular product. So the founders of RPT, again, Mark Humayun, Dennis Clegg, and David Hinton, who have been giving us a tremendous amount of effort and time in helping us start up RPT. There's myself also, I'm a former CSO and Senior Vice President of Regenerative Medicine in Sharon, and now acting as the President and CSO for the organization. We also have Katie Spink acting as our COO. So I think I'll just finish up here by saying that we're really looking at putting all of the various elements that are going to be required to commercialize this product. We have product attributes that support the pathology observed in geographic atrophy. We're looking at supporting broad market distribution too, with the fact that these can be cryopreserved. We're looking at our clinical targets include both orphan and large indications. We have a broad IP portfolio. The cGMP manufacturing is an established process right now. It's being transferred to the City of Hope, and we're looking at imminently starting our qualification and engineering runs. The preclinical studies support our target product claims so far. We're looking at starting an IND-enabling studies early next year. Again, we've had our pre-IND meeting. The funding we have in place right now for supporting the project through to the IND, through, again, the great support of CERN, and then the team experienced in ophthalmology and in fact having a very successful track record in developing products to commercialization.